a powerful technique in molecular biology. We'll discuss various techniques, such as and more. Join us as we unravel the two-dimensional difference gel electrophoresis, 2D DGE, is a technique that allows simultaneous separation and quantification of multiple protein samples. It uses isoelectric focusing, IEF, and SDS-PAGE to visualize and compare protein expression differences in samples, aiding in disease and experimental studies. In the isoelectric focusing, IEF, section of the video, I will explain how this gel electrophoresis technique separates proteins based on their isoelectric point, pi. I will discuss the pH gradient gel or immobilized pH gradient, IPG, strip used, and how proteins migrate towards the region where their charge is zero, resulting in their separation. I will also highlight how EF is commonly used as the first dimension in two-dimensional gel electrophoresis for high-resolution protein analysis. Protein identification after gel electrophoresis is crucial in proteomics. Mass spectrometry, MS, is a commonly employed method, where proteins are digested into peptides, ionized, and analyzed by MZ. The resulting mass spectra are compared to identify proteins, even in complex samples. Phosphoproteins, proteins with phosphorylated residues, play crucial roles in cellular signaling and regulation. Phosphorylation events can modulate protein function, localization, and interactions. Gel electrophoresis can be modified to specifically detect and analyze phosphoproteins. Phosphoprotein gel electrophoresis techniques involve the incorporation of metal ions or specific stains that selectively bind to phosphorylated residues, allowing for their detection after electrophoretic separation. These techniques enable the study of phosphorylation dynamics and identification of phosphorylated proteins in various biological samples. BN-PAGE is a gel electrophoresis technique that analyzes protein complexes in their natural state. Unlike traditional SDS-PAGE, it maintains protein structure and interactions. A mild detergent is used to solubilize proteins while preserving their non-covalent interactions. This technique separates protein complexes based on size and composition, aiding in the study of multi-protein complexes involved in cellular processes like oxidative phosphorylation. Understanding protein-protein interactions is crucial for deciphering cellular processes and signaling pathways. Cross-linking gel electrophoresis techniques enable the study of protein-protein interactions by covalently capturing interacting proteins within a complex or within close proximity. In these techniques, a chemical cross-linker is used to covalently bind interacting proteins, which can then be visualized and analyzed by gel electrophoresis. Cross-linking gel electrophoresis provides valuable insight into protein complexes, their stoichiometry, and the dynamics of protein-protein interactions. Glycoproteins, proteins with attached carbohydrate moieties, play critical roles in various biological processes and diseases. Gel electrophoresis techniques can be adapted to specifically analyze and characterize glycoproteins. Glycoprotein gel electrophoresis involves the use of lectins, proteins that bind to specific carbohydrate structures, as probes for the detection and separation of glycoproteins. By exploiting lectin-carbohydrate interactions, researchers can identify and explore changes in glycoprotein expression and modifications in different biological samples. In TGGE, a gel with a temperature gradient is used to separate and analyze DNA, RNA, or proteins based on their melting temperatures. This technique is valuable for studying SNPs, mutation screening, and protein stability and conformational changes. Chemical cross-linking gel electrophoresis is a technique used to probe the proximity and interactions between biomolecules, such as proteins or nucleic acids. In this technique, a cross-linking agent is added to a sample, which covalently links interacting biomolecules. After cross-linking, gel electrophoresis is performed to separate the cross-linked complexes based on their size. This technique provides valuable insights into the structure and organization of biomolecular complexes and the mapping of protein-protein or protein-nucleic acid interfaces. Protein complexes play critical roles in cellular functions and signaling pathways. Native gel electrophoresis techniques enable the separation and analysis of protein complexes while preserving their native structures and interactions. In native gel electrophoresis, proteins are separated based on their sizes and charges without the use of denaturing agents, such as SDS. This technique allows for the visualization and characterization of protein complexes, their stoichiometry, and their interactions with other molecules. Quantitative proteomics measures protein expression levels in different samples using gel-based methods like gel electrophoresis. Variations in protein levels are detected by the intensity of bands or spots on the gel. Techniques like 2D DGE, and SILIC provide accurate quantification, aiding the identification of important proteins in biological processes and diseases. High-resolution gel electrophoresis techniques, such as capillary gel electrophoresis, CGE, offer enhanced separation capabilities for smaller molecules, including nucleic acids, peptides, and small proteins. CGE utilizes narrow-bore capillaries filled with a separation matrix to separate analytes based on their charges and/or sizes. This technique provides high-resolution separation and increased sensitivity, making it a valuable tool in fields such as DNA sequencing, proteomics, and metabolomics. CGE has revolutionized the analysis of small biomolecules, enabling precise quantification and identification in complex biological samples. Biology and proteomics. It separates and analyzes techniques like SDS-PAGE, 2D DGA, expression, interactions, modifications, and in gel electrophoresis will advance biological and